Okay, so the tonsils are lymphoid organs that basically form a ring in the back of the mouth or around the throat, sometimes called the ring of Waldeyer. And it's just an extra protective layer because there's a lot of air turnover as air comes into the lungs and out. So it's just a way to protect our bodies from things that are coming in and out through the airway. As far as their naming, they're named based on their location. So we have the palatine tonsils, the lingual tonsil, the pharyngeal tonsil, which is sometimes called the adenoids, as well as the tubal tonsils. And again, they're going to contain immune cells such as lymphocytes that are going to protect us against pathogens that make their way to the posterior portions of the oral cavity. And of course, inflammation of the tonsils is known as tonsillitis. It used to be believed that the tonsils really didn't have much of a function, but come to find out they actually do. So here you can see some of these tonsils. Here's the pharyngeal tonsil, the palatine tonsils, lingual tonsils. And again, this is just an extra protective mechanism that our body has due to that high turnover of air coming in and out of the mouth. Peyer's patches, or aggregated lymphoid nodules, are basically these little bundles of lymphoid tissue that are found mostly in the distal ileum. Not a whole lot is known about Peyer's patches, but they are protective against bacteria that may be able to get through the intestinal wall and set up shop and cause an infection. They generate memory T cells. We'll talk about memory T cells a little bit later, but basically memory T cells are necessary for our long-term immunity. The appendix, which is basically hangs off the first part of the large intestine, which is connected to the distal ileum of the small intestine, and it also has very similar aggregated lymphoid nodules, and these also play a role in the immune response. So the appendix also has a likely role, whereas in the past it was believed to be kind of a non-essential vestigial organ, but that's not the case. So here you can see these are these little Peyer's patches, these little lymphatic nodules in the distal ileum. Okay, let's talk more about the immune system. So the immune system is a functional system that contains various cells, chemicals, structures. The definition immunity is just simply resistance to disease. And we have two main divisions of immunity. We have innate immunity and adaptive immunity. Innate immunity is innate. We have it from birth. This is very fast-acting, quickly responding, and it's non-specific. We also don't really have memory when we talk about innate immunity. Adaptive immunity is more slow developing, but it's specific, and we generate memory. And we're going to see, even though we have these two divisions, they do talk to each other. So let's talk about innate immunity first. So the innate immune response is basically going to provide our first and second lines of defense for the body. The first line is going to be skin, mucous membranes, and the secretions of these membranes. So a couple of examples of the first line of defense. The acidity of the skin. So the skin is acidic by pH, and this is going to inhibit a lot of microbial growth for the most part, because most microbes like to live more in a neutral pH. The stomach. So the mucosa of the stomach will secrete hydrochloric acid. Hydrochloric acid in the stomach keeps the pH around 2, which is lethal to most microbes. However, it's been discovered that approximately 200 or so species of bacteria can still grow in the stomach. The most famous is probably H. pylori. Saliva and tears and other secretions in the body contain lysozyme. Lysozyme will break down the bacterial cell wall, specifically gram-positive bacteria. Mucus is very important in the digestive tracts, the respiratory tracts, because it will trap and sweep away or eventually move out microbes that get in. So again, some other examples here. So the pH of the stomach, as we talked about lysozyme, the skin. So the skin, of course, is a physical barrier, but it's also acidic. A rapid pH change. So as we go from the stomach into the small intestine, we have a very rapid increase in pH, which can also be deadly to a lot of microorganisms. Normal flora that inhabit inside and outside the body also prevent the growth of bad things, which is important. Flushing of the urinary tract, so it's very important to flush the urinary tract to wash away any potential pathogens. Okay, when the first line of defense is breached, then we will encounter the second line of defense. And the second line of defense is really going to be cells and chemicals. So a couple of cells that we've talked about before, macrophages and neutrophils, which are the primary phagocytes in the body, will consume any pathogens that get to the tissues. Natural killer cells, or NK cells, are actually a type of lymphocyte, and I kind of call them the police of the blood. So you can find them hanging out in the blood and lymph, and they will kill not only infected cells, like virally infected cells, but also cancer cells. So they're important for killing cancer cells. Antipathogenic proteins, again, these are just some of the chemicals, and we're going to talk about some of them. So the first type of antipathogenic proteins are interferons, IFNs. And when you hear interferon, the first thing I want you to think is defense against viruses. Okay? They're also important for other aspects of the immune system, like bacteria, parasites, and tumor cells. And there are many different types of these. But basically, our cells, our host cells, such as lymphocytes, macrophages, fibroblasts, that have been infected, will produce these interferons. And interferons are kind of like almost like a smoke signal to other cells. And they will reach surrounding cells and cause the production of antimicrobial proteins. And also proteins that inhibit viral replication if the host cell is infected by a virus. Interferons also important for mobilizing and activating other immune cells. So basically, if you think of this as a smoke signal, it's going to bring other immune cells to the site. It's going to bring other fighters in. So this is a really simple picture, but I like it a lot. So here is a cell that's infected with a virus. This cell is going to produce these interferons, this smoke signal, and it's going to signal neighboring cells to do some function. So for example, it will cause the production of antiviral proteins. It will signal surrounding cells to undergo apoptosis, which is programmed cell death. And that's important because if this cell essentially commits cell suicide, these viruses can't infect it, and therefore the virus can't spread. And it will also activate other immune cells. So some of these other proteins, the complement system. So the complement system is the system of plasma proteins that are very important for innate immunity. And basically there are 3 pathways that can activate this system, but the big takeaway is what this system does and what these proteins do. So for one, these proteins will coat microbes. They will basically act as little flags around these microbes. That process is called opsonization. And this flagging of the microbes will basically signal to the phagocytes, hey, come eat me. These complement proteins will also poke little holes in the membrane of certain microbes. These holes or these pores are known as the membrane attack complex, the MAC, and this will cause these cells to lyse and die. These complement proteins are also inflammatory, and with that they will recruit other cells like basophils and mast cells and neutrophils to areas of inflammation. And this is a lot more detail than you need to know, but if you ever take an immunology course, you will learn this in great detail. It's lots of fun. Okay, so the inflammatory response. This is kind of a hallmark of innate immunity. So inflammation, or the inflammatory response, is triggered when there is some damage to our body's tissue. This can be due to an injury. This can be due to pathogen. Just any sort of damage to our tissues. And there are 4 cardinal signs, sometimes 5 if the damage is great enough. But the 4 cardinal signs are rubor, calor, tumor, or dolor, better known as redness, heat, swelling, and pain. So the inflammatory response will generally progress in the following steps. So the first thing you have is you have damage to cells. And when you have damage to cells, they will release inflammatory chemicals into the extracellular fluid. Some of these chemicals include histamine, prostaglandins, kinins, which are important for pain, cytokines, which are chemical messengers between cells of the immune system. And these cells will cause the blood vessels, the capillaries, to become more permeable, and also cause them to vasodilate. And that's why you start to see swelling. That's why you see redness. The fluid will leave these capillaries, and in this fluid you have clotting factors, you have antibodies, which is very important for healing. The complement system that we just talked about will be activated, and we will also have white blood cells come into the site. And this is a really important component of the inflammatory response, the entering of white blood cells into the inflammatory site. So if somebody has an infection, like a bacterial infection, you're going to have an increase in neutrophils, you're going to have an increase in monocytes, and this can cause leukocytosis, which is a high number of white blood cells. Now, damaged endothelial cells will produce these little adhesion molecules, and these adhesion molecules are little stoppers for neutrophils and eventually monocytes. Okay, so it helps the white blood cells kind of stick to that endothelial surface. This process is called margination. And then the phagocytes, so the neutrophils and the monocytes, will actually leave the blood and enter the inflammatory site. This process is called diapedesis. So margination is basically the sticking of the white blood cells to the endothelial wall. Diapedesis is the blood cell, or the white blood cells actually leaving the blood. Now, there are inflammatory chemicals at this site that act as chemotactic agents. Basically, all that means is these chemicals attract these white blood cells to the site. That's how they know where to go. And then once the inflammatory response is over, we can have healing. So this is kind of showing this process a little bit. So you have some sort of damage. You have these chemotactic chemicals that are attracting white blood cells to the site. White blood cells will diapedese out of the blood and into the site. Fever. So fever in general is a systemic response to pathogens. The substances that actually cause fever are known as pyrogens. Pyrogens can come from our own cells, like our own immune cells. Those are known as endogenous pyrogens, or they can come from foreign cells or foreign viruses, and those are known as exogenous pyrogens. Normally, fevers cause no problem at all. They might be uncomfortable, but they serve a very important function. Okay, they increase metabolic rate in pretty much every cell, and this helps to not only fight pathogens, but also repair tissue. It increases cell-mediated immunity, so it helps our T cells work better. It helps interferons work better, which we just talked about. But of course, a very high fever is dangerous, but they're also exceptionally rare. Okay, let's move into the adaptive arm of the immune system. And the adaptive immune system is specific, and this will be our third line of defense. Okay, the adaptive immune system will recognize specific pathogens or foreign substances known as antigens. And this works systemically as opposed to being restricted to a specific infection site, which is kind of the case for innate immunity. So after initial exposure to a pathogen or an antigen of some sort, the adaptive immune system is able to mount an even stronger response against subsequent exposures. Now, our immune system has to recognize our own tissues. This is known as self-tolerance. And that's why, I talked about how the training of these blood cells, of these immune cells like B cells and T cells, is so stringent. It's because they need to be able to recognize things that are bad and leave our tissues alone. The breakdown of self-tolerance really is the basis for autoimmunity. Okay, so the adaptive immune response involves two separate but united responses. And this is going to involve T cells and B cells, or T and B lymphocytes. So cell-mediated immunity involves defense of the body by T cells, and this can be either direct or indirect. Humoral immunity, also known as antibody-mediated immunity, involves defense through antibodies. Antibodies are produced by plasma cells. Plasma cells are mature B cells. Remember, all lymphocytes, all blood cells are produced in the red bone marrow, but the T cells will mature in the thymus, which was one of our primary lymphoid organs, and B cells will mature in the bone marrow, the other primary lymphoid organ. Okay, antigens. Antigens are basically substances that can cause an adaptive immune response. They are things that can cause the production of antibodies. And really, almost anything could be an antigen. When we're talking about pathogens, parts of pathogens, foods, certain proteins like, in peanuts and seafood and things like that, viral proteins, almost anything can be an antigen. Now, all of our cells have antigens, and one of those antigens are the MHC antigens, also known as major histocompatibility complex antigens. And these are on the surface of most of our body cells. And there are actually more than two classes of MHC, but we're just going to talk about two. We have MHC class 1 and MHC class 2. MHC class 1 is found on all nucleated cells in our body, with a couple of exceptions, but for the most part, all cells in our body that have a nucleus. And then MHC class 2 is found on immunocompetent cells, specifically cells known as antigen-presenting cells. So there are three of them, dendritic cells, macrophages, and B cells. Those cells will have MHC class 2. Now, each of us has unique MHC, with the exception of identical twins. And this is really the basis for rejection when it comes to grafts and, transplants. Okay, antigen-presenting cells are cells that present antigens. And basically what they will do is they will engulf an antigen, they will break that antigen apart inside the cell, and then present those antigens on the surface. And there are three antigen-presenting cells, three professional antigen-presenting cells: dendritic cells, macrophages, and B cells. And again, this is going to involve MHC, specifically MHC class 2. So here is kind of how the process of antigen presentation works. An antigen is going to be ingested, it's going to be taken into one of these cells, and it's going to be broken apart into fragments. This MHC class 2, is going to be synthesized at the same time. It could be MHC class 1, but MHC class 2 is specific to these cells. We're going to have vesicles inside the cells that contain those digested fragments, as well as vesicles that contain the MHC. And the antigen fragments will bind to the MHC. Okay, this MHC antigen complex will actually reach the surface of the cell, and that's how it's going to present this antigen. This is quite a bit of detail here, but basically we have ingestion of antigens that's going to be dissolved and broken apart, and it's going to be assembled with MHC class 2, and then this is going to travel to the surface of the cell, and there's our little fragment of the antigen that's being presented. So you're probably wondering, well, what is this being presented to? And really it's going to be presented to T cells. Okay, so let's talk about cell-mediated immunity in T cells. And there are three main types of T cells. We have cytotoxic T cells, we have helper T cells, and we have memory T cells, which are really just a subset of helper T cells. Cytotoxic T cells, also known as CD8 positive T cells because they have a CD8 receptor on their surface. And these are going to be activated by body cells that have presented antigens by that MHC class 1 molecule, or the MHC class 1 antigen. So remember, all of our nucleated cells in the body have MHC class 1. Okay, so when this is presented, when an antigen is presented on MHC class 1, it's going to activate cytotoxic T cells. And what's going to happen is when it recognizes that, the T cell is going to bind and destroy these cells that have the MHC class 1. And they do that by releasing enzymes, specifically perforin and granzymes. So basically, the cytotoxic T cell will kind of dock right here. So here's our MHC class 1. This was presenting the antigen fragment. The cytotoxic T cell will dock on this cell. It's going to recognize this specific antigen. It's going to release perforin, which causes pores in this cell. And then it's going to release granzymes, which will enter this cell and kill this cell. Okay, very effective against virally infected cells as well as tumor cells. So just imagine this. Imagine this cell is a respiratory epithelial cell. and it is infected with the influenza virus. This cell is going to digest that viral protein. It's going to put it on MHC class 1, and it's going to present it to this cytotoxic T cell. The cytotoxic T cell will dock and then eventually kill this respiratory epithelial cell. Helper T cells. Helper T cells are very, very important. These are known as CD4 positive T cells because they have CD4 receptors on their surface. But they do so much. Helper T cells are going to activate B cells to become plasma cells. They are going to produce a number of signaling molecules which will activate other T cells, cause the division of T cells. I mean, we could really talk all day about helper T cells. Memory T cells. Memory T cells are really a type of helper T cell that are able to recognize previously encountered antigens. And the purpose of memory T cells is to allow for a faster response when you are subsequently infected with something. So this little note down here, don't pay too much attention to this. Just talking about co-stimulation, we're not really going to get into that in much detail. This is just showing some of the functions of T cells, helper T cells. Helper T cells do a lot. They really do. So I'm not going to get into a ton of detail about it, but just know helper T cells are very, very important. Okay, now let's talk about humoral immunity. So humoral immunity involves plasma cells. Plasma cells are B cells that have matured, and plasma cells will produce 5 antibodies. Okay. We also have memory B cells. Memory B cells, again, just like memory T cells, will be ready to respond during a subsequent exposure to an antigen. So let's talk about the 5 antibodies. There are really only 4 of them that are important for us, and the way to remember them is GAME, G-A-M-E. So IgG, it is the most abundant antibody in serum, in the blood, so you might want to add that in. It's not the most abundant antibody in the body, but it is the most abundant in the blood. This is the only one that can cross the placenta, which is important. So it gives the fetus some protection from the mother. It's going to be able to activate the complement system, which we talked about briefly. IgA is going to be the most abundant antibody in the body, and this is going to be found mostly in mucous membranes. So this provides us with mucosal immunity. IgM is the first antibody produced by plasma cells. So all plasma cells will produce IgM first. And then with the help of helper T cells can actually produce different types down the road. IgM will also activate complement along with IgG. IgD, nobody really knows exactly what this guy's all about. It's found on the surface of B cells, so a lot of people think it's involved with B cell activation. And then IgE, you find IgE on the surface of mast cells and basophils, so I want you to think of IgE as being involved with allergic reactions. So here you can kind of see these proteins, they're really glycoproteins. So here's IgG, IgE, IgD, IgM, IgA. So some of the functions of antibodies, other than the ones we just mentioned, is they neutralize antigens. IgG will be very important for that. They activate complement, specifically IgG and IgM. They enhance phagocytosis. IgG can do this pretty well. They can cause agglutination of pathogens. They can cause pathogens to clump together. IgM is particularly good at this. And they also immobilize bacteria. So IgG, IgM, IgA all can do that pretty well. And again, just showing some of the functions of these antibodies. Okay, so one of the hallmarks of adaptive immunity is the idea of memory, or immunological memory. And this is basically having antibodies and lymphocytes which arose from cells that encountered an antigen. Okay, but in order for this to happen, in order to develop memory, you have to have a primary response. You have to have a primary encounter of your adaptive immune system and an antigen. So the primary response is when lymphocytes encounter an antigen for the first time. And what you would see if you were to take somebody's titers is you wouldn't have many antibodies for a few days, and then you would start to see an increase in IgM because IgM is the first one produced. And then after that, you'll have an increase in IgG. And that's because you have helper T cells that help the plasma cells produce a different type of antibody. Now, the secondary response, this is a subsequent encounter with that same antigen. So what you have is you have a much more rapid increase in antibody levels, specifically IgG, because we don't have to produce IgM first because this is a subsequent encounter. Okay, so if you ever hear the whole thing, you can't catch the same cold twice, that's kind of true. And that's because of the secondary response. So here's a primary immune response. This is, you can think of this as IgM. And then here's a secondary response. You have a rapid increase in antibodies. You can think of this as IgG. And you'll still have some new cells becoming activated, so you can think of this one as IgM. But the big takeaway is you have a primary response. In the primary response, we make memory cells. That way, when we encounter the antigen again, we remember it and we can mount a much stronger immune response. This is why boosters are often needed for certain vaccines. There are really four types of adaptive immunity. We can break this down into naturally acquired and artificially acquired. So we have active and passive types of both. So we have naturally acquired active immunity. This is when, this is basically you getting sick. Okay, you're catching something from nature very naturally. Okay. We also have naturally acquired passive immunity, and this is when a mother is going to pass antibodies through the placenta or through breast milk. Okay, so it's naturally acquired because it's coming from a natural source, but it's passive because the fetus or the infant is just receiving antibodies. It doesn't have to actively make them. Then we have artificially acquired active immunity. This is basically vaccination. So it's artificially acquired. These vaccines are made in a lab, but it's active immunity because your body has to actively mount the immune response. And then we have artificially acquired passive immunity. This is when you have preformed antibodies that have been artificially developed. Maybe they were artificially stimulated in a horse or something, so you get these antibodies from horse serum, and then you inject it into somebody who is not immune to whatever disease you're talking about. So the last couple slides, I just want to talk about HIV and AIDS. So acquired immunodeficiency syndrome, or AIDS, is caused by the human immunodeficiency virus, or HIV. So AIDS is the syndrome, HIV is the virus. This can be transmitted through blood, through sexual contact, sexual fluids, as well as mother to fetus. HIV primarily infects helper T cells, but also infects macrophages and dendritic cells. And basically what HIV does is it doesn't really kill the T cells, it just makes them non-functional, and eventually we exhaust kind of our supply of T cells. And what you do is you basically wipe out cell-mediated immunity. So one entire arm of adaptive immunity, and that's very dangerous. So this is going to make these patients very susceptible to infections and cancers, specifically infections and cancers that really nobody else gets. Once you reach a certain level of T cells, when your T cells are low enough, the disease is officially classified as AIDS. Without treatment, life expectancy is about 10 years, again, give or take a couple of years. But some people can live normal lives if treatment is started early. And here's many details about HIV and AIDS. Again, this sort of pathology stuff would be more for bonus material on the exam. So if you have any questions, please ask. Please bring your questions to class on Thursday, and we can go over any of these questions you may have.